Thank you very much for coming. So, yeah, we at Exscientia, we do believe that all drugs will be designed using artificial intelligence by 2030, and we believe that for two broad reasons. One, because we and some others have shown that it is possible, and two, because we have to do it as a field. Drug discovery is too slow, too costly, and just is not an economically viable industry at the moment. And so what I'm going to talk to you today about is, why is this so important? Why is it so difficult? And what is our approach to solving this problem? So, as I think most of you all know, drug discovery is far too expensive. It takes 30 years to go from a biological idea to a drug, and it takes 10 years to discover a drug itself. It costs billions and billions of dollars. It's not an economically viable industry. And why is this a problem? It's a massive problem because there are 10,000 diseases, and most of them are treated or are largely untreated or uncured. And so what we see is that the failure rate is just so colossally high that the industry is economically very challenging. Increasing the probability of success from 4% to just 10% would increase the ROI by 600%. And so there is a massive moral and economic argument that says that we need to improve the efficiency of the process. At Exscientia, we've been using AI and generative approaches for over a decade to discover drugs, and have already shown that you can increase the probability of success, you can increase the efficiency by 70%, you can increase the economic viability by 80%. And so we believe that now is the time to use AI to discover drugs. What have we done? So we've produced tangible results. We've developed 8 development candidates. So we did 3 with our DSP partnership in the early days of Exscientia. And then we have 5 drug candidates, either in clinical trials or moving towards clinical trials. One of which is partnered with, one of which has been taken by BMS, that's the PKC theta inhibitor. And the other 4 we either fully own or part own. In discovering each of these development candidates, what we have done is learn about new challenges in drug discovery, and we've been able to tackle them and then encode them into our system. So as an example, for the PKC theta project, we needed to develop the potency and the selectivity in particular against other kinases, such that it could be administered on a daily basis for a long period of time. And that required us to understand kinase selectivity, feed that into our systems, and then design compounds against that. Likewise, LSD1, we needed to develop a CNS-penetrant molecule with a half-life that was appropriate for safety. And again, that required us to tackle further challenges, and I'll talk about that later on in the talk. What do we do at Exscientia? So small molecule design is a component of what we do, but we go all the way through from target selection. So we have our own AI methods for finding targets for reading literature. Then small molecule design, and we also are building a biologics design capability. We have an automation capability for generating high quality data. We have a patient tissue screening and selection capability for generating biomarkers. And we're also thinking about adaptive clinical trials and how we can optimize clinical trial design to make that process more efficient. So we're thinking all the way across the drug discovery landscape. But I'm today going to talk to you about small molecule design. So at Exscientia, we talk about encoding and automating drug discovery. And I'm going to talk about why we need to do that, and also why drug discovery is so hard. So the first reason drug discovery is so hard is because the chemical space is so large. There are 10 to the 60 drug-like small molecules that you could make. That is an astronomically enormous number. It's more than the atoms in the entire solar system. It is not possible, even computationally, to brute force a space that large. The largest virtual libraries are 10 to the 12, 10 to the 13, even now. That is not even a drop in the ocean compared to 10 to the 60. Even 10 to the 59, right? Even 10 to the 59 may not be big enough to brute force. The data are incomplete, so we do not believe that this is a big data problem because the data are so small and they're so heterogeneous, and data gathering is so slow and expensive. It costs $10,000 to make and test each compound as an average. So generating millions or billions of data points is incredibly expensive, and even then is only a small drop in the ocean of the overall matrix. The third reason drug discovery is so hard is because it is a multi-dimensional optimization problem. That means that each time you improve one property, you will quite often remove or reduce one of the other properties. So they are often competing, often diametrically opposed, and is a problem that is very well suited to artificial intelligence approaches, but incredibly challenging to get right, in particular when you need to predict each of those individual endpoints. So we use active learning very across our drug discovery processes. And this is in order to maximize the learning with the resources available. So this is a cartoon landscape of a drug discovery process, where each of the crevices are bits of knowledge that we have about the overall landscape. If you imagine this overall landscape is almost infinite, spanning across. Every time we design a compound and we make it, we open up one of those valleys. We understand a bit more about it, and we can find the ones that we find most promising. We can hone in on those, and then we can use that knowledge to find the drug candidate that fits the properties that we want to observe. So this is a process of exploration first, and then exploitation to find the compound that we want to see. We believe that drug discovery is a learning problem, not a screening problem, and we think about it with this particular analogy. If you think of a word, you could have a word in the English language, say there's 600,000 words it possibly could be. You could have 20 yes or no questions or 20,000 guesses. If you have 20,000 guesses, chances are that you will fail 97% of the time. If you ask 20 perfect questions, the chances are that you will succeed. If they're exactly perfect, then you will succeed. You will find the answer. In terms of information theory, once you've got to the 10th question, you're spanning 1,000 possibilities. After 15 questions, your probability of success is again only 3%. But then once you've got to the 20th question, you've been able to span that knowledge base. I think there's two really important points from this. Firstly, the quality of the questions that you ask is crucial. If you ask very bad questions, that's as bad as having 20 guesses and you've got almost no chance. The second point is that the learning is exponential. So early on in the project or early on in the series of questions that you're asking, the amount that you know about the information space is still pretty small. And then as you get towards the end of the process, the amount that you know becomes greater and greater and greater. And so you need to be patient and sit with the process. That's why when we talk about encoding and automating, there's a massive cultural challenge as part of this. So why do we encode? We encode to teach and to have a system that can learn. If you don't encode the drug discovery process, if every time you do a drug discovery program, you start from scratch, there's no way that you can learn about what the best way to tackle that problem is. Why do we automate? We automate for quality, so we have increased reproducibility. This is what people learned during the Industrial Revolution. And we automate for speed. If we can automate processes, we can move more quickly, we can learn more quickly. But a lot of this is culturally quite challenging because it hits against the human element of how we currently do drug design. And one of the major things that we've learned at Exscientia is that we need to tackle those cultural challenges and we need to keep moving forward with them and understand that the process is going to look different with drug discovery using AI. The process of doing AI drug discovery is different from the process of doing human-driven, human-led medchem. What does it look like? So we start off with our project objectives, our available data, and our proprietary starting points. And then we move through this cycle of building models, performing generative design to build populations of compounds, active learning to select from those populations, chemical synthesis, biological testing, and then moving through that process again. We do 20 to 30 compounds per design cycle, which is mostly driven by synthetic expediency. That number may change in time as we enable automation to change that number. And we also have 10 to 15 design cycles from hit to drug candidate. So we have these 10 to 15 different cycles of design, asking different questions along the way. What does that look like in more detail? So on the far left, we have what we call our target product profile. This is what we need a drug to look like. This means potency, selectivity, stability, the ability of the molecule to be absorbed into the body. We can then encode that into a multi-parameter optimization function, where we match each of those experimental endpoints that we want to achieve with a prediction or a property. And we can then set our generative algorithms against that multi-parameter optimization. We then make and test those compounds, and we assess our progress using a system called Merit, which enables us to objectively and quantitatively see how far away we are from the goal. It normalizes our compounds and combines all properties together using a geometric mean, such that every compound is scored between 0 and 1. And typically on projects, what we find is we have this initial exploration phase where the compounds are scoring quite lowly, 0.1, 0.2. And then over time, we learn into the project, and we have more of an exploitation phase where the compounds are all scoring quite well. This enables us to understand when we've succeeded on a project, when we've achieved our goal, and when we have not. Because this is one of the key causes of inefficiency in the process. Cool. So what does generative design look like? So this is a real generative design run from our A2A program. These are the compounds that we made and designed. And what we're doing here is we're keeping the core of the molecule static, which is often what we will use as a constraint. And then we are designing novel molecules that fit into the protein binding pocket and scoring them against not just how well they fit into that pocket, but also how well they would conform to other properties as part of our NPOs, so how much they would be stable and how well they would be CNS penetrant, for example. And then we optimize and we find the best compound in the population, and we move that forward or the best compounds in the population and move that forward into active learning. Here we're using a multitude of different methods, including gated graph neural network methods, RNN methods, as well as more classical kind of evolutionary approaches as well. Second, I'll talk about active learning. So here we find that active learning enables us to get to creative breakthroughs in projects. In this case, it came to a counterintuitive selection against the preconceptions and the dogma of a human, how a human would think about a project. So here we're trying to optimize against potency and CNS. And the higher the number in each case, the better the compound. Each of the shapes on that diagram is a different compound. So any of the shapes towards the top right are in theory the best compounds. Any of the squares are the compounds that we selected using active learning. And so what you can see is rather than just picking all of the compounds towards the top right, as a human might be inclined to do, the active learning algorithm picks 2 of the worst compounds in this selection. They're still predicted to be somewhat potent, somewhat CNS penetrant, but they're 2 of the worst compounds. The reason it picks those is because they asked a different question of the data. They asked a question of the data about a more simple molecule with less elaboration and with a key structural motif missing. These were then synthesized and tested, and what they did is they enabled a breakthrough in the project, and they enabled us to move forward and actually eventually led to the lead compound from which the candidate was optimized. So counterintuitive. So this is what we do. We take all types of data, we apply generative design, we add our physics platform and our ML models, active learning, and then we're developing automation that will enable us to automate the chemistry and the biology on top of that. So I'm going to talk to you about our MALT1 program. This was the first time we used physics as part of our AI design approach. And the reason we did that is to solve a problem. So we wanted a selective and potent compound that solved some of the phys-chem problems that we saw in other compounds that address this target. We wanted to bind to the allosteric binding pocket. And early on in the project, we found that the pocket definition, the static pocket definition, was not appropriate for design. It was not designing active compounds. And so we used molecular dynamics to understand the pocket better and to inform our generative constraints. We then, and this is what every design cycle at Exscientia looks like, we evolved compounds into that better characterized pocket using our evolutionary or generative algorithms. We used models to predict the properties as part of an NPO. And then we used active learning to select the compounds. And this is what that looks like. So what we previously were seeing was a pocket that was long and flat. And then as you see in the molecular dynamics simulations, as I'm showing here, the pocket grows towards the top. And that was a key part that only when we knew that would our algorithms be able to design against it effectively to design potent compounds. And in the end, here's our candidate profile. So our compound is on the far right. And again, the key focus here is not on any one property. So each of the rows is a different property, and it's color-coded by whether it meets the criteria, almost meets the criteria with a minor deviation, or if there's a major deviation. Each of the columns is a different compound from a different competitor. What we see is our compound not only has the potency in the top few rows, but also doesn't have a UGT1A liability, which is an off-target that we didn't want to be hitting, causes liver tox, and has improved solubility over many of the competitor compounds. So we have a more balanced compound, a compound where we've been able to design using physics that we've embedded into our platform. This then led us to build our own automated physics capability. So what we believe is that physics is just another form of data that we can use to feed our AI algorithms. So we use it for potency prediction. We use it for pose prediction. On the far right, I can show our RBFE method, which we've developed, our in-house RBFE, relative binding free energy method, which we've developed. Benchmarked against other commercial competitors, we show our method is as good or better than commercial competitors. The reason we built our own method, though, was to enable us to have automation and scalability from the off, and also for us to be able to develop our own methods that will enhance either our ability to perform certain transformations, or most recently, we're developing our own QML, our own quantum mechanics machine learning methods that will enable us to use quantum mechanical level accuracy in our simulations through using machine learning potentials. One really core component of all of this is domain-specific data preparation. So here we're talking about three-dimensional data. So this is proteins and ligands in complex with each other through a method known as X-ray crystallography primarily. And we need to be able to prepare data in 3 dimensions in an appropriate way for automation. This requires a huge amount of domain expertise, both from people with a structural bioinformatics understanding, so people who really deeply understand structural data, or from an experimental background, or people with a molecular dynamics background, or more recently, people with a diffusion modeling background will enable them to generate protein structures using deep learning methods. This requires those groups to all work together, and this is far from a solved problem. We require this scale to be able to incorporate it into our process, but we also need this automation for us to understand whether our methods are becoming better or not. Often what we've seen in previous sort of increases in the field is what they've actually been demonstrating is manipulation of the underlying data itself. So we need automated approaches to be able to generate the data to input into our physics-based methods. Another key component of all of this is open source. So this may not be a massive news to this group, but open source software is hugely important for all of this, and is something that has historically often been underutilized within drug discovery. So academic research, which is where a lot of the cutting-edge breakthroughs are happening in physics-based modeling, has largely transitioned to an open source model. And we can see that with just the sheer amount of the tools that are being developed. But secondly, it's very clearly a massively powerful innovation engine, and we believe that it's a really, really powerful way for us to move science forward, which is what we care about at Exscientia. So we've been working a lot with our open source tool developers, and we've built our physics platform on the back of the open source community. So what have I told you today? We believe that all drugs should be, need to be, and will be designed using AI by 2030. We've been able to demonstrate that we can deliver 8 precision design development candidates that have solved unmet medical needs and have required us to develop novel capabilities to solve each one. 6 of the projects have been in-licensed by partners, showing that pharma also understands the value of these compounds. And through doing this, we've been able to show a 70% reduction in discovery time from target ID to candidate, and 80% improved capital efficiency, which is both massively important for us to be able to solve the massive unmet need that exists. Thank you so much for your time, and I'd be very happy to ask questions after the talk.